All right, great. Well, thank you so much for having me today. It's going to be a fun evening, I hope, with lots of talk about ticks and how you can prevent tick bites as well as the disease that are transmitted by those ticks. As Tony said, I'm the director of the Midwest Center of Excellence for Vector Borne Disease, and that is a grant-funded organization that's regional. And the whole mission of the Midwest Center of Excellence is to try to find ways to reduce the risk of ticks and the diseases that they transmit, as well as mosquitoes, but we won't have time to talk about the mosquitoes tonight. So I was thinking about this and trying to come up with some sort of an image that maybe all of you would remember and take home. And this is what I managed to put together from the AI Google Gemini. So I asked it to make me a maple syrup lollipop with a tick embedded in it. And I hope all of you out there are kind of groaning and going, ew, because sometimes that kind of response is just what you need to remember that this is the time of year when you need to start really being careful and watching out for those deer ticks. So fix this image in your mind, and then we're going to go ahead now and talk a little bit about several different topics. So I wanted to start a bit with the biology, just talking about the ticks that are common human biters in Wisconsin, and then some aspects of their biology that's really important to really, I think, understanding um, what they feed on and what they transmit and what kinds of things you need to be looking for when you're trying to protect yourself. And then many of you provided a lot of questions to Tony in advance of our meeting this evening. And I did look at those today and think about trying to include some material that would be responsive to your questions. Some of those questions really focused on the changes in populations of ticks that we're seeing. So I will try to talk a little bit about some of that tonight. And then I also want to talk about some of the trends we're seeing with tick-borne disease case numbers in our state, because we are a hot spot in the US for tick-borne disease. And then again, I'll get to this issue of what we can do to try to prevent tick bites. So I just starting here with this idea of uh, how important it is to know the kinds of ticks that are here and what kinds of things you need to be watching out for. So my slide title is Know Your Ticks. These are the two ticks that are the human biting species in our state. We probably have about 14 species, but most of them are so tightly associated with their animal hosts that we don't ever see them feeding on people. But probably all of you are familiar with these two, the American dog tick and then the black-legged tick. There is actually also another tick I just wanted to mention briefly here at the beginning, and this is a tick that's called the Lone Star tick. And often I find that many people in our state have already heard about the Lone Star tick, even though this is not a tick that is established here. So it's really rare. We don't have any populations that we're aware of this tick that can make it through our winters, unlike the American dog tick and the black-legged tick, which have no trouble getting through our winters. But the Lone Star tick actually can't do that yet. But what we always see with Lone Star ticks is that they feed on birds in the immature stages. And so in this spring migration period, that nymphal stage that you see here on the screen is feeding on birds. It gets brought up here, it finishes its blood meal, drops off into the ground, and turns into the adult. And then that's where sometimes people in Wisconsin are actually encountering, usually it's the adult female. And they do send us pictures of those and they, report their travel history. So we know that they didn't go down to Arkansas and pick up the tick, but they definitely do get introduced, but it's just really rare. I think we get less than 20 reports every year. But I will say, um, even somebody like me, out in the woods, one time I was way up in northern Wisconsin, uh, literally miles and miles and miles out in the woods away from anybody else. And I was sampling for black-legged ticks, put down my blanket drag, and I picked it up, and there was a Lone Star tick on it. So what are the chances of that, right? That the one person in the state who's really interested in these ticks would pick one up. So they're not, I say rare, but that definitely not impossible that you could encounter one too. And if you do, we're very interested in gathering that information. So please don't hesitate to reach out. You'll see my contact information will come up later in the presentation. Okay, so I said these are the two common human biting ticks in Wisconsin. And the first one I said was the American dog tick. I want to also comment that many people in Wisconsin call this a wood tick, and it's the same thing. Those are exactly the same thing. The other thing is sometimes people get this tick, and because of that white that you see there on the plate on the back, they actually think that it's this tick, that Lone Star tick, but no, see that white spot on the back of the adult female? It's really distinct looking compared to how that wood tick looks. So then the other thing I wanted to comment is you've been hearing me say black-legged tick in relationship to this second tick on your screen. That's our deer tick. That's what most people in Wisconsin still call that. So you're going to hear me probably say deer tick or black-legged tick interchangeably throughout this talk. So it's really, again, it's the same thing, two different common names for the same species. Now, importantly, it's this one, the deer tick, that is associated with 99.999% of all the human disease transmission in our state. So that's the one I'm really going to focus about for the rest of my talk. It is true, though, that both of these are found in forested habitats. And that may be kind of important because I think sometimes what can happen is that people pick up the wood tick in these same habitat and then maybe later they get sick. And what they saw was the wood tick. And so they associate the being sick with the wood tick, when actually, because the two kinds of ticks are in the same habitat, they could very well have picked up a deer tick in the same place, gotten sick as a result of the bite of that tick, and then really just misunderstood, what was the source of their infection. In this kind of place, the deer ticks really have to have this very high humidity environment in the forest, so you rarely see them outside of these kinds of spaces. The wood ticks much more frequently are going to be out in like open meadows in a bright sunny kind of space, but they'll also be, as I said, in spaces like this. Okay, so now next I want to share a little bit about the tick life cycle. And one thing that I always find really striking is that because I also work on mosquitoes, that the ticks have such a long life cycle. Like with an average mosquito, adult mosquito is going to live for a couple weeks, maybe a month. It's a really short life cycle. The ticks, though, from the egg stage all the way through to a female laying another batch of eggs, it is 2 to 3 years. So it's a long time. And that plays out in a number of different important ways, but we'll describe those in a minute. First, I wanted to show you, though, in this figure, those eggs up in the upper right corner that have been laid by that blood-fed female. So that is a female tick there. She's kind of collapsing now as all the eggs have been produced. Those eggs, like 3 weeks to a month later, usually will hatch into this little six-legged larva. The larvae are really tiny, very hard to see. You can also see that it's kind of a light color there, and that light color means they're sometimes a bit transparent and a little hard to see on any background, really. If the larva is successful in finding a host and getting a blood meal, then after 3 to 4 weeks, it will molt into this next stage, the nymphal stage, which is at the top there with that other arrow pointing to it. And so nymphal stage you see is quite a bit bigger, and now it has 8 legs. If it's successful in finding a host and getting a blood meal, then again, after about 3 weeks to a month, it will turn into an adult. And it can either be, again, that adult female, I don't know if you can see my pointer, but right here, and then the male on the other side. And we can't tell which it's going to be just by looking at the nymph. They don't have any external manifestations. So the only way you can tell if this is a male nymph or a female is by doing a genetic test. One thing I want you to notice on this slide that's super important is the difference in size between the adult female and the nymph. And you're seeing that also on the finger over here. Both of those two stages are associated with transmission of most of the important diseases in our state. Larvae can transmit one or two things, but they're extraordinarily rare. So it's really the nymphs and the adult females that we worry about. And those adult females are big enough so that it's a lot more likely that you'll find them and get them off. And they're also active, most active, like now. And so they're not quite as out there and active during the period when most people are actually getting exposed and sick, but the nymphs are. So we think that the nymphs are a really important driver of disease, and this being so small and so hard to find is a part of all that. So, okay, so that's that one. Oh, I was going to show you this. This is a much reviled, a very hated photograph. It was part of an ad campaign that the CDC put out just to get people thinking more about the size of the nymphs of the deer tick so that they would have the right search image when they're trying to check themselves and make sure they didn't pick anything up. So it's a lemon poppy seed muffin, and they glued a bunch of deer tick nymphs onto the top of it. So if you focus in real closely, you might be able to see them, but if not, here you can see them a little bit better. There are five of them glued onto the top of that muffin. So again, hopefully all this is giving you a little bit of a sense of what is the size of the thing you ought to be looking for as we get more into the summer months. Now, some questions that people had shared already before our meeting tonight was about the activity patterns of these ticks in our state. And so we've got this nice little graphic that I thought I'd share with you just to show you what's out and when they're out. And maybe we'll just start with April since that's where we are right now. So if you look at that part of the pie chart, you'll see that that's an adult tick. So really right now in April, that's all we're seeing. It's actually kind of it's a banner moment if we get a nymph in April because it happens so rarely. It's possible, but it's going to be like one. They're just not out there in any numbers yet. So right now is the peak of the adult time. So this is exactly when we would go out and try to collect a lot of those if we wanted to get them for, say, looking at infection prevalence or something like that. The adults will continue to be active in May, and then they start to tail down. So we're at peak right now for adults. In May, they'll still be out, but there'll be less of them. In June, same thing, still out. We'll get them, but there won't be as many. And then by the time we get to mid-July, they're gone. We simply do not see adult deer ticks in mid-July or August. We start to see a few again in September, and then that October, November is usually a very active period for them. But of course, October, November, not so many people outside. The kids are back in school, so again, although they can certainly be infected with the pathogens, just the amount of disease associated with them is a lot less. And then the next important thing is to look at when those nymphs are out. Oh, I know what I was going to say, though. I forgot this because somebody had asked about could they be active all year round? And yes, in fact, if you get a warm day and it's 40 degrees, usually is what we think. It can even be a little bit of the high 30s, but high 30s in a more open place where there's a little bit of sun getting in and warming it up, right? So, yes, we absolutely get them in December. I think our first ones this year in February when we had a couple of warm days. Don't think it ever got warm enough in January, but it has in other years. So anytime in the winter that it warms up above 40 and you're in the right place, you could indeed encounter one of these adult female deer ticks. So you want to keep actively checking yourself even during the depths of winter in Wisconsin. All right, so then this next key piece is about those nymphs because they start to get active in May next month. They will peak in June or the first couple weeks in July. The highest numbers are usually the last 2 weeks in June, but there's been a few years where that got pushed back to July. So lots of nymphs out at that point. They're still there in August and tailing off. It's not like the numbers that we see in June and July, but you can definitely find a few, same in September, but not many. And then after that, we just don't see those again. And then larvae usually start to be abundant about a week or two after the nymphs really start showing up. And then we'll see those June, July, and August. And after that, we don't usually see them for the rest of the year. They come at this point, they've tried to find blood. They're molting into nymphs or they're staying as blood-fed larvae until the spring when they turn into the next stage. So that's the 2 to 3 year life history or life cycle of the deer tick in our state and kind of when you can expect to see them out and about. And remember that I said that the nymphs are important because of the disease incidence. You can really see it in this map. This is from our own Department of Health Services, where you got, yes, we have cases that are reported in January, February, March for sure, maybe reflecting things that were in the fall, perhaps November, maybe December, and then that doesn't get reported till January. But most of the cases coincide with when the nymphal activity is occurring. So I want to talk next briefly about a little bit of the blood feeding biology and especially this issue of how long do they have to be on you in order to transmit a pathogen to you. I'm sharing with you here a really nice graphic that, well, nice, I guess, is depends on your perspective, but I'm showing you a graphic of how the adult female progresses through her feeding. She's usually on the host from 7 days to 10 days. And you can see, in the first few days, she's still pretty small. She's just beginning to feed by a day and a half after attachment, just starting to swell up a little bit on day two, starts to go faster on day three, and then a four and five, you just see by day seven she's enormous, almost unrecognizable actually as a deer tick. That is a heck of a physiological challenge too for her to have to grow that big in that short a time. Um, this is coming from an organization called the Tick Encounter Resource from a colleague of mine at the University of Rhode Island, and he does a great job of collecting a lot of information about all things tick related. So if you're interested in learning more at his site, you can actually click and watch like a little movie of them growing. So you could take a look at that. The other thing that I think is important here to mention is that they get that blood from, well, if you're unlucky, you or your pet, but usually it's from white-tailed deer. And it can also be from other like carnivores, bigger animals that are out there. But weirdly, we just never see adult deer ticks on any of the small mammals. So we do a ton of work looking at things like mice and chipmunks and sometimes squirrels and shrews, we just don't see adults on those animals. So something about they must encounter them once in a while and they just reject it and don't get on for the 7 to 10 days that they need for the blood meal. So that's an interesting biological problem. And then this time to transmission element, I wanted to mention again. Here's a little nymph that was feeding on me actually for 4 days. So even though I was very careful and did my tick check, I totally missed it. And then by the time I found it, I was like 2 days from going on a vacation. And I thought, oh boy, really don't want to be on vacation and sick. So I contacted my doctor and they will often give you a preventative medicine. So it's a single dose of doxycycline. Especially that'll work if you get it off within 17 hours or if you get the drug within 72 hours after you've taken the tick off. That's the time when infection is most likely to just be getting going, moving out from the place of the tick bite. So it our laboratory studies suggest that it takes more than 36 hours of attachment for a nymph to start transmitting the Lyme spirochete. You're often going to see advice that will say 24 hours. And I think we do that to be careful, right? Because there's always going to be situations where it's just a little bit different for someone. So we're saying between that 24 to 48 hours is probably what's most usual. So if you can get them off within the first day, then the chances that they uh, infected you with the Lyme disease spirochete is very low. And the lab work suggests that again, it has to move out of the gut and get into the salivary glands. And it takes it a while through those early stages of feeding before that can happen. So I want to talk a little bit about diseases now. And I do like to start this part of the talk by just a reference to Wisconsin as a high incidence state for Lyme disease. So in the map here, you see data from 2023. I think the 2024 might be the most recent data, and it looks worse than this even a little bit. But you can see us, and then you see this eastern seaboard with just every one of those is a case, and it just blends all into one big mass of blue in many places now. Another important aspect on this slide is just mentioning that the CDC has done a series of studies over the last 5, 10 years to evaluate how many people in the US are actually getting diagnosed and treated for Lyme disease. It used to be that they could only report out what was reported to them from each of the states. And there was just a sense that those reports were not capturing the intensity of the problem. And indeed, when they went in and looked at insurance claims for treatment and diagnosis, they realized that probably what they had in their records was about a tenfold underestimate, perhaps. So instead of 30,000 cases, which is what we used to tell people, maybe more like 300,000 cases, which is a pretty big discrepancy. And again, we're one of the hot spots in the country for this disease. This is just reported cases, 6,400 and something in 2024. If CDC's estimate is accurate for our state, this is probably more like 64,000 cases instead of 6,400. And again, they can only do what they get from the counties, right? So I think they've recently just changed at our state level their method of surveillance so that now they work directly with the diagnostic companies to get that information. The other thing I wanted to share here is people had asked about change over time in some of those pre-session questions. And you notice that early on in the early 1990s to about 2000, it was kind of stable, and it was maybe 400 or 500 cases were reported each year. And then these reported cases just started to go up and up and up. And, now it's like 10 times higher by the time you get to 2010, and then it's just continued to increase ever since. And acknowledge that there have been changes in the way they report, but still, I do believe that this is an accurate trend, that we are seeing increases here for a lot of reasons that I'll share with you next. Um, One of those reasons is you could really see it here when you look at the geography of Lyme disease risk in our state over the last, oh gosh, what's this now, 35 years, something like that. You see, here's where most of the Lyme was back in the early 1990s. And then by the late 1990s, you could see that it has begun to really expand down into the southwestern part of the state. By the mid 2000s, now you've got a much more intense situation again, kind of all over that west central, maybe not the bottommost southern part of our state, but certainly the rest of that tier, and not the easternmost part of our state either. And then this last map is 2008 to 2010. And again, just a real intensification of the problem, with much of the east and the south just not seeing as much. And so that was 2010. So here's one of the more recent maps. And now you can see again that same area, right? Most of the north, except it's now expanded over into this northeastern quadrant where we didn't have much before. Door County is beginning to experience some problems that it hadn't had before. You still see a lot of these more southern and eastern counties with less of a problem. So they still have it, but it's just not as intense. And that probably relates to the forested habitat need. So if you go into these counties and you find the right kind of habitat with the forest, you are going to find the ticks now. It's just not as widespread as it would be in many of these other counties, the right habitat. So again, to think about the patterns and trends, that was disease, and what's happened with the ticks has really tracked right along with that. So this is like the very first records from our state were from an animal where a black-legged tick was collected, and it languished hidden in our insect museum for a long time. That was in 1958, and I just found it a few years ago. But In addition to that, about 1965, there were some forestry workers in Lincoln County who discovered some ticks that they didn't recognize, had not seen before. Now, forestry, so they're outside all the time. So they recognized this was something new. They sent them down to my predecessor here at UW-Madison, and he ID'd them as black-legged ticks. And then he went and did some collections up in northern Wisconsin and also down along the eastern part of the state. He didn't find much like south, like Jackson County and Monroe, like down in that area, he really didn't find any, but he did find a few other locations up north. And that was the mid 60s. Just wasn't very interesting to people at that time because it was like, okay, big deal. There's a new tick in Wisconsin. But then we had the whole Lyme disease story begin to emerge in the late 1970s and the early 1980s. And that got people interested, knowing that that tick did occur here, and doing some surveys to see just how extensive the distribution was. And that work was carried out by taking advantage of the relationship with deer and the gun deer season, because you could go in to the registration stations and take a look at all the animals that were coming through and get an estimate of what percentage of those animals actually had deer ticks attached to them. So in 1981 here, you could really see in the eastern part of the state, you could look at a lot of deer and you just didn't see any ticks attached to them, really. It was very hard, maybe one. I ran this survey in 94, and still it was getting more intense down the center of the state, like from Eagle River on down to Mazomanie, but not much again still over here in the eastern part of the state. And then the last one I did was in 2009. And now, finally, by this time, you're really starting to see it pick up in the southern part of the state, but also most of those locations in the east, too. It's not a lot necessarily compared to over in the northwestern part or the central western part, but definitely they're there where it doesn't really look like they had been. So the ticks have been moving across the state. And we go out and collect ticks in other ways besides just deer check, like by going to all the counties, the right kind of habitat, and we do that blanket dragging across the landscape. Um, and now we can say that every single county in Wisconsin has an established population of these ticks. So it's not necessarily that the whole county is infested, but if there's the right kind of habitat, there's at least one park or other could be private land where we've been able to find a established population. I do have one on here that says recorded instead of established, but that one we flipped last year. So we have them all now red. Part of the reasons for this change in our state over the last 30, 40 years has to do with longer term patterns related to what happened to our forests with the big deforestation at the turn of the century. So you see how the virgin forest looked at the top of this map, and then at the very bottom in 1926, most of that forest was gone. These guys need forests, so that was important. And then we also had this issue of a massive loss of much of the deer population. So you see again, right around 1900, the deer population had just been hunted to almost to extinction, and was to extinction in some places. And then that pressure was released and it began to rebound pretty quickly. So you see by mid-century, 1950s here, it was really coming up. So the recovery of that, the regrowth of some of the forest, probably saw a little bit of this change in the deer tick populations has to do with changing temperatures. All of that has contributed to some of the patterns that we've seen over time. Okay, so that's a little bit about change. I also wanted to mention some of these other tick transmitted diseases that are associated with the different ticks. The black-legged tick is the most important one, where we have 7 different pathogens that it's known to transmit. I say 7 because there's actually 2 different species that cause the Lyme borreliosis, but one of them is very rare. We have another bacterial pathogen called anaplasmosis, and we have about 780 cases of that, I think the last time I checked for last year. That one, sometimes people don't get a rash, right? Like the bullseye rash is a little bit, not always there, but when it is, it's pretty diagnostic of the Lyme borreliosis. But you can get sick with anaplasmosis without getting borreliosis and not really recognize it in the same way you would if you had the bullseye's rash. So I do ask people to be careful about if they get a tick bite, even if they get a negative test for Lyme, it doesn't mean they don't have something like anaplasma. Um, and let me think, let's see. Yeah, I had a friend, had a colleague, and that's exactly what happened to him. He was really sick. They gave him the antibiotic. They did the test for the Lyme. He came back negative. They took him off the antibiotic. And it took several weeks before him being sick all over again, before they finally did a test and realized that he had anaplasma. And then they put him back on the antibiotic and he was fine. So those are the two most common. We have these rare ones that you see at the bottom, including babesiosis, which is like a malaria type of a parasite instead of a bacteria or a virus. We don't have a lot of cases of babesia, but it's going up. So again, it's just something to be aware of. And then we have these really rare things, which, yes, they occur, not anything that I would worry about. But if you want to look more into the patterns related to those, go to the Wisconsin DHS, and they have all of the maps and the history and everything that you could want to know about these different pathogens. We have a few things that are transmitted by the Lone Star tick, but a lot of that is travel. Somebody went to Arkansas, picked up a tick, got sick, came home and got diagnosed here. These diagnoses are county of residence as opposed to county of exposure. So, and then last of all, the wood tick, which same problem. We have some people who get this spotted fever group rickettsia that's associated with this, but most of them have travel history. So can't attribute that to infection here in the state. Okay, people also asked about the Lyme disease vaccine and is there any hope of one or where are we with that? There is a candidate that's already in phase 3 trials, so that's pretty far along in terms of evaluating how well it works. And that one might be approved as early as 2027. I did just go to a meeting where they said there were some problems with the trial in that not enough people got Lyme disease for them to be able to make conclusions in the control group. So there may be some delay. It depends on whether the FDA will accept the kind of evidence that they have. But it's coming. And so that may be something for you to think about. Last time I checked this, it was about 70% effective. I think you would have to have annual vaccination, and maybe there's more than one shot to get you boosted right at the beginning. But there is something coming. Just remember, though, that even if you get a Lyme vaccine, that will protect you from what's by far the most common tick-borne disease. It's not going to protect you from anaplasma. So you'd still want to be taking precautions and just, being careful if you did get ill after a tick bite. Okay, this was a recent article by one of the most knowledgeable people in the country about ticks and tick-borne disease. And the title of his article is Why Are the Ticks Winning and What Can We Do About It? So I want to talk about what we can do about it next. And part of what the Midwest Center of Excellence for Vector-Borne Disease works on is these very operational or applied kinds of topics. So we really want to understand so that we can make good recommendations how things related to personal prevention measures actually work. How do-it-yourself methods for homeowners might work on their properties. What about private pest managers who are willing to come and treat your property? How well does that actually work? And then we're thinking a lot about whether some kind of neighborhood level programs where people banded together, whether you might not get a better result than just trying to do it on a small property based uh, methodology. And this is really based on our like surrounding states. A lot of them have these tax funded mosquito control districts, tons of them in Chicago. There's a big one over in Minneapolis, St. Paul. There's a lot in Michigan. We don't have any of those here in Wisconsin, but it's a model that we could think about maybe for some areas for trying to reduce the risk of tick-borne disease. And then the last thing that we're interested in, and I've just started working on, is strategies for forested areas. We've done a little bit of work on like mowing trail side and some recreational areas, only to find, unfortunately, that it didn't really help or make any difference in the ticks in those areas. But we're starting to look at a few other things that I'll share in just a couple minutes. I want to talk first, though, about some of the things that we do know work to reduce ticks abundance on properties. And these are using these synthetic acaricides to treat what we call the ecotone. So it's really just that little zone right between where the property backs up to the forest and the lawn is what we're trying to hit, because that's where the tick densities tend to be the highest and where the risk of encounter is also the highest. And so these work. We have seen anything from like 95% suppression to a little lower, down more down the 60s and 70s. Sometimes it's a little challenging to compare to the controls. You get a lot of variability, but something like that will we get suppression in the zone that gets treated really well with things like some of the pyrethroids that we've looked at. So that was a liquid application. We've also done some work with granular products and used like a little hopper, almost like a fertilizer applier, right? Or a seeder that you might use in your lawn. And it just throws out a little bit of those granules, or again, right at that edge. That seems to work really well too. We like that. We did a project with homeowners last summer in a neighborhood up around the Wisconsin Dells. And the good thing about it is it's a little easier to get the right amount of stuff down with this than it is with the aqueous preparations. So this will work as well. The one thing about these kinds of applications, though, is you do have to be very careful to read the label and do it right. This to me looks really a little unnerving because notice that he's coming right up on where all these flowers are, and that's absolutely not what you want to do, because that's when you would have a hit on your pollinator and your beneficials. So it's important to put down the right amounts to not overdo it and to really pay attention to where you're allowed to put them. Proximity to water is another thing they have to be very careful about with these pesticides. And of course, not everybody wants to use the synthetics. And so there are a lot of natural product based acaricides that people have been interested in and that are on the market now. And so these belong to a group of chemicals that are have very limited regulation by the feds, the EPA, because they're thought to be minimal risk because they're made of plant essential oils. They really don't require any efficacy data. So we went out and we pulled together about 16 different products, and we worked with them quite a bit to try to figure out what is the right way to test them. But ultimately, we landed on this, well, if you dip the ticks in the products and it doesn't kill them, then it's really unlikely that you're going to be able to put them in a sprayer and go around the edge of your yard and get any death. So we started with all these products just by doing this immersion assay. And what you can see in this graph is like more half of them, they don't even kill 25% of the ticks when they're fully immersed in the stuff, right? So that's not good. We did find a couple that hit this standard of 80% mortality. So there were a couple down here on the end. And so we decided we would take those to a field trial. So here's our field plots. You can see us laying them out. These are going to get sprayed, and then we'll know where they are, and we'll come back and drag for ticks over the next few weeks. And this is kind of what that looks like. So we go out to the field, and the blue ones never get treatment. So we're dragging them looking for the time when the tick populations are starting to come up. And then right before we're going to do the application, which is here on the dotted line, we drag everything. So the controls as well as the plots that are going to get the products. Then we do the application, and then we come back every week for a month and we look. And unfortunately, there was just no sign that those products were really making any difference. When we have seen a difference with one other product that we're taking to the field again this year, it's like this first week or day after we put the stuff down, then we can sometimes see it. But here we're not even getting that. This is all noise, really, because it's so variable. So we put down the second treatment. same thing. We just really didn't see anything. So right now, I do not have a natural product that I could recommend to you for a property-based kind of a treatment. We're still working on that. We have one still in our back pocket we're hopeful might be better, and that one will get tested this year. And I also mentioned earlier that we're going to be doing some work with forest management. We just really started with this, getting going. We did a little bit with the mowing. We're now interested in this idea of prescribed burns and how that might affect tick density. This isn't my work. This is, you see the PMID number down in the corner. That'll take you to the paper of the people who did this evaluation. And they basically took every study they could find on controlled burns and what that did. And I mean, okay, decreased density, but it's not much. 25 to 22 ticks per 100 meters is just not sufficient to make any difference, in my opinion. They did report that burn intensity probably matters, and so that's something to think about for further studies. A little bit of evidence from just timber harvesting, again, not real strong. Better on the invasive species front. But still, 25 to 15, it'd have to be part of an integrated program. I mean, you have less chance if there's only 15 of encountering one, but still it's not low enough really to be very satisfying. One of those studies actually was some work that we did in the UW Arboretum where we were putting a couple things together. We were trying to target these mice that are so important in feeding the larval ticks and then producing those infected nymphs. And we combine that with a invasive species removal in some of the plots. So we're testing the tick tube, which actually has a permethrin treated cotton in it. And the mice come and get the cotton and they take it back to the nest and then they get the permethrin on their fur. And then any larvae that try to feed on them are not successful in completing that meal. So now remember the life cycle. The larvae would be feeding on mice this year, and then they would turn into the infected nymphs that are out questing next spring, right? So we expect there to be a 1 year lag from putting this in place. And we did see actually about an average of 53% reductions in the questing nymphs in those places that got the tick tube. So those are forested plots. It could be you could do something like that plus maybe one of the synthetic acaricides or uh, for us, the vegetation removal didn't really seem to improve the use of the tick tubes. It actually reduced the how effective that seemed to be a little bit. So don't yet have exactly the perfect IPM set of things that we can do. Other problem is that it's not just mice. So this technique only targets the mice. Nothing else takes the cotton. And we know that chipmunks can be really important in some situations. And we also know that shrews can be really important in feeding a lot of ticks in some situations. So we have this community ecology problem of trying to figure out methods that will target each of these hosts if we want to take a host targeted approach. And we're not quite there yet. We're working on a few things, but we don't have something yet. Okay, so I want to tell you now how we protect our field teams from getting sick. So we
Hi
send it.
there. How's
Hello.
it going?
Hi. So I want to tell you how we protect our field teams from getting sick. In like 12, 15 years of sending people out to some of the tickiest parts of Wisconsin, we've only ever had one person who ever got ill. So pretty proud of that. I think the things we do actually work well to protect them. So one is this message that you've heard a lot, do a frequent tick check when you're out. Look for those ticks and check your pants and check your shirt. This is a former graduate student of mine, Jordan Manley, who's being my model for this. And as I said, the sooner you get them off, the better, the less likely that it will transmit disease to you. I do recommend if you find one, you might think about just marking that day. So if you get sick, you'll remember when you had the tick. And also if you save it and throw it in the freezer, you can take it to the doctor with you and they can verify that you actually have a tick bite, in the right frame from when you got ill. We have done a bit of work since this is such a cheap and easy thing for people to do, and it's just a constant part of all the recommendations that we make. We've done a little bit of work to try to figure out whether people are actually even doing tick checks. And we did a big survey in our area and found that a lot of people reported they were doing thorough tick checks most of the time or always, which was awesome. Really happy about that. Unfortunately, though, we also did some work trying to figure out if people even know what they're looking for by putting some embedded in resin and then asking people which ones of these are the ticks. Some of them are actually other kinds of insects. And unfortunately, most people would recognize the adults, and weirdly, they get that Lone Star tick way more frequently than they actually recognize the deer tick. But they don't recognize, like, here's the little nymph in the middle. They really infrequently know that that's what they're supposed to be looking for. So that's a problem when we say do a frequent tick check, as we do here in this piece of advice. One other way that we've been kind of looking at that is to use department store mannequins. Actually, we have a whole family of mannequins now that we take out to events and that we also use for research. So the mannequins get ticks glued onto them. So you see the little one on the collar, and then here's another one on the shirt in different places. And then we put people in a tent with the mannequin and a sheet of paper that has a outline of the mannequins, and they are asked to put a little sticker in every place where they think they see a tick. And here is the outcome of that. We have all different stages and everything. And the smaller the spot, the fewer the people actually were able to find it. So you see ticks 1 and 4 here. Very few people. Tick 2 on the back over here. Very few people are finding those. Tick 11 was under the shirt, so not many people found that. 5 people didn't find that either. So all in all, people only found about 47% of all the ticks that were on a mannequin dressed in black and about 70% on white, but that still left 30% they didn't find. And most of the ones they didn't find were on the lower half of the body. So we've been thinking about that. There's probably some mobility issues. Can we come up with some tools that might make it easier for people to spot them? Maybe some reflectivity with a flashlight, or maybe you could use your phone on a selfie stick and take a picture or something. But it's really clear that it's hard. And thinking about how tiny those nymphs are, just really hard to do a good tick check. So what else? I mean, we also recommend that you shower within a couple of hours of being outside and use a scrubby because that will help dislodge any ticks that aren't feeding or just barely feeding. Use a repellent. EPA has a number of repellents that they recommend, and so I always say go look at their website. I like the DEET and the picaridin ones at a 20% concentration, but there are others. If you're not comfortable using DEET, there's lemon eucalyptus, and there are even some of the essential oils here, but they don't last very long. So you'd want to check and think about how long you have to be outside before I think you'd want to go to that. All my people have to wear long pants and they got to tuck their pants in their boots. When we get back, we don't always change because sometimes we're not in a situation where we have ready access to laundry. So if we can put them in the dryer though at high temperature before we go out again the next day, then we always do that, and that will kill them. Any ticks that are trying to hitch a ride. You can treat your clothing. So there's a compound called permethrin in this. You don't want to put it on your skin. It's a repellent, but it's not a skin repellent. It's a repellent to put on your pants. And so we always do that. This is a requirement of everybody on our field team. I go to St. Vincent de Paul. I buy some cheap clothes. I get my repellent. I wait for a sunny day in my driveway. I do a little treat on the front, a little treat on the back. I hang them up and let them dry, and then they're safe to wear. Not everybody wants to do this either, put the permethrin on their clothes. And I just found this study about a lemon eucalyptus oil that looked like it was as effective as DEET, actually, as a fabric repellent, and it lasted for several days. So that one isn't available, but it may be something to watch for in the future. They've tested it. It'll go through some more iterations, but that is something that's kind of promising. The tucking your pants, that's important. I do want to say that I make my team wear these rubber knee boots. So let me show you why. Watch that tick. Remember I told you they get on close to the bottom, he falls off. because they're always questing low. They're not up very high at all, these nymphal stages. So this student of mine was in the Navy. I just wanted to show you that second one. It does the same thing. He fells off. And yeah, here's my Navy entomologist with his more camo boots. There, same thing. So we do think that these boots might be helpful. That they just don't like the texture of it. They're not getting the signal they need, and so they don't continue walking up and in bed. So this is my summary of all the things that I recommend that you do, just like our team does, that will help to protect you from getting any of the tick-borne diseases. You can also protect your pets. Number of ways to do that. I just comment here because people sometimes say they think frontline doesn't work because they see the ticks crawling on the pet. Well, that's not how it works. Actually, the ticks have to have attached to imbibe the frontline. And so it's not surprising that if you're using something like that, they'll still get on your animal. There are some other things that kill them a little quicker than like the frontline product does. So lots of opportunities, different kinds of things that you might want to consider. I like the vet school at Cornell for this kind of information. So I've put a link down here if you wanted to go check that out. Okay, good. Then the last thing I want to talk about was our tool called the Tick app. And this is a research tool that we developed to try to understand more about what people are doing when they're being exposed to ticks. So I'm just going to tell you right off the bat that if you decide you want to help us out by participating in our Tick app project, it is a research project. So you'll start by having to fill out informed consent in the app and then doing a baseline survey that will ask questions about like behaviors in your kind of home environment and things like that. But once you're in the app, then we have all kinds of features for users that are, I think, valuable. One is a tick activity, and that will tell you like what's happening right now where you live. So in Dane County, for example, when this screenshot was taken, it was high nymph time, and there's a little picture to just remind you about how small those nymphs are. We also have information about how to get ticks off. What's the right methodology for that, as well as a little video there. All the things we've talked about for tick prevention, we just reiterate. We have Tick 101 because a lot of times people ask us questions, and so we have tried to bring them all together and put them in the app so they're right there and ready to go if you have them. And then we have a couple of ways that people can report their tick encounters. And one of those is this feature called Report a Tick. So let's say you get a tick on you and you want to know what it is. You can take a picture, you can upload the picture, and then my team will identify what that tick is and send you a message right back and let you know. And we can often tell how long it's been feeding, which again is helpful to you because if it's a flat tick, you don't have to worry about it. If it's a tick that's been feeding for a while, then that's the point where we might suggest that you talk to your healthcare provider. So the Tick app, here's the website for the Tick app, and it's free. It's on Google Play and it's on the Apple App Store. And that is my last slide. So if you have questions for me, in the chat. Maybe we have a minute or two to take those questions. Otherwise, Tony can provide those to me, and I'm happy to try to do any additional answers, post our meeting today. But thank you so much for being here.
Yeah, thank you so much, Susan. That was a great presentation, a lot of really good information. Yeah, and we do have quite a few questions in the chat right now. So maybe we can take a couple of minutes and run through some of those questions, and then I appreciate the offer to provide a written response to some of the questions that we don't get to tonight. But I will just point everybody again to that chat feature. I have been entering links in for some of the websites, including the Tick app that Susan has been talking about. And as I share the recording of this session, I will share the links that are shared in the chat as well. So we will get all of that out in a summary email, hopefully by the end of the week. Maybe it'll be early next week. But with that said, we can dive right into some of the questions. So we got a couple of people asking about what actually eats ticks in the wild.
Yeah, it's a great question. I think there's some evidence that a few birds will eat them, not enough to make a dent in the population. And there used to be some studies like guinea hens, for example. People were trying to use those to control. If they get on things like raccoons, the raccoons will groom them off and eat them that way. There's probably some predatory beetles and ants that'll eat them, but it's just, it's not enough. Whatever out there does eat them, it's not enough to really do much damage to the population.
Okay. Um, so do you have a sense of what percent of the black-legged ticks carry Lyme's disease in Wisconsin?
Yes. Yes, that's a lot of the kind of work we do is to look at the prevalence of the infections in the ticks. And pretty much throughout the state, it's between 20 and 25% in the nymphs and between 40 and 60% in the adult ticks. So,
Okay.
on average, about half of the adults have a females, adult females. The males don't usually bite, which is why I usually just talk about the females. Um,
hmm
yeah, so it's a lot, right? One out of every four or five if it's a nymph, and one out of every two.
Okay. Yeah. Wow. And you mentioned a little bit about the kind of necessary time that it takes to actually transmit the Lyme's infection.
Yeah.
If you could just reiterate that point, and then is there a difference between that transmission time between nymphs and adults?
Okay, yeah, sure. For the nymphs, which is where several studies have been done in a laboratory setting, so you can be very controlled about it. It's pretty clear they need 36 hours or more before they start transmitting spirochetes. And again, our advice is 24 hours, get it off within 24 hours, and you got very little chance of it actually transmitting that pathogen to you. Um, the adults, to be honest with you, there just isn't a whole lot of studies that have looked at that question. There is one that suggested that it might take longer actually for the adult, so maybe more like 3 or 4 days before it's starting to transmit, but I just don't think it's all that clear. So I'm confident about the nymphs. I'm less confident about the adults.
Interesting. Yeah, and we really appreciate the research backed information that you're sharing here, and a lot more questions coming in. So there's one about infection called Bartonella. Is that a tick-borne disease that you're aware of? B
Well,
A
I've
R
heard,
T O N.
of course, I've heard of Bartonella, yes. But the evidence that Bartonella is being transmitted by black-legged ticks is very weak. So right now, I don't think so. What I have seen was an interesting study where a lot of people who had had a tick bite had their blood taken, and they looked very carefully for any novel or interesting or unusual kind of patterns in that sample that they had. And they did actually find a new pathogen, something that had never been identified in people before. They found no sign of Bartonella in any of those samples. And it was like 20,000. It was a huge sample size. So I just don't think the evidence is strong in any way that Bartonella is being tick transmitted.
Okay, interesting. And we've got a couple of questions about some of the um, chemical applications, kind of the landscape applications for tick control. And
Sure.
if you could just talk a little bit about, potential impacts on other insects or wildlife.
Mhm.
There's some questions about other
Yeah. Yeah.
insects, and then also about the the species that are eating the insects, so the birds, or if we're even on the like the permethrin that we're
On the
the
mice.
mice are. Yeah, is there
Hmm. Yeah.
impacts on, say, if a cat would get that mouse?
Yeah, yeah, yeah. Well, so first about the area, the barrier treatments, the idea there is that you don't treat a very big swath, right? You're just trying to get that edge habitat where the ticks are the most abundant. So imagine like a few meters wide, like a pretty wide mulch barrier, maybe something like that you could visualize. So yes, it's not going to just kill ticks in that area where you put that treatment down. It will kill other things that enter that area that are already present in that area. So if you get ants like moving across that barrier, it's going to get the ants. Absolutely. That's why we try to keep it real skinny though, like just that one little stretch right along the edge with the forest rather than doing the whole entire yard, right? So that's why that's the recommendation.
Mm-hmm.
And your second question about the animals is an interesting one. The amount of permethrin that you're getting on the fur is pretty low. I don't think anybody has ever seen an issue with like a feral cat, like getting a mouse, and then having a problem as a result of the exposure to permethrin. That would be on the fur because I think it's a pretty limited amount of that compound. But that is definitely something that's interesting. It's a great question.
And it's definitely on, people's minds with some of the issues with rodenticides that are used for controlling,
Yes,
those
I've
mice.
been thinking
And
about those rodenticides myself a lot, having had a little Cooper's hawk on our street and then a great horned owl that died. And we were all just kind of sure that, oh, somebody's using a warfarin-like compound, and we're seeing a
hmm
problem in our raptors. So absolutely
hmm
agree. To be sensitive to those things is really important.
We've got a couple of folks thanking you in the chat for the presentation and answering the questions and just being available here. We are 5 minutes past the hour, so just want to again thank you for your time. We got a couple of questions about uh, ticks as they're questing, and they
Mm,
say if the vegetation's 2 feet high, will they go all the way to the ends of that
mm.
vegetation?
Yeah,
And
great question.
there's often this perception about ticks dropping out of trees. Could you just talk a little
Yeah,
bit more about how they're actually moving from
sure,
the
sure.
landscape onto people?
Yeah, I really like that question. Thank you. So I have been in like waist high grass and picked up adult deer ticks before. I was up in the Flambeau National Forest and walking down one of the trails where the grass had gotten pretty high. And so the adults, I do think, will go up higher, much higher than the nymphs. The nymphs don't ever seem to get that high. They stay much lower. Um, the and the larvae even lower than the nymphs. The issue of them falling out of trees is an interesting one. To the best of my knowledge, they do not crawl up and jump on you out of trees or fall on you out of the trees. They're really reliant on you brushing up against them, actually. But I will share
hmm
one story that made me kind of pause on that. I was at a lake up at a Boy Scout camp looking for ticks, and down by the lake, there were these maybe 8 foot tall short trees or shrubs or something. And I went under one of the trees and like jogged it a little bit. And I had my drag with me just in the right position. And a couple of ticks actually did fall off of these little branches right onto the right onto the drag. So I think that they had actually crawled up the tree. Not that they're really jumping on you, but maybe when deer brushed up against them in that area, they were getting on the animals that way. That's the only example, though, in many, many years of doing this work that made me wonder, well, maybe there's some situations where you get a little shrubby thing and they kind of fall out of that. But
hmm Got time for two more quick ones?
sure.
Okay. So there's one about possums and rumor that possums are eating hundreds of ticks a day. Is that true or not? We already talked about this, but just specifically with the possums.
Yeah, yeah. Possums is kind of an interesting story, and people got excited about that. But unfortunately, I think it was really not very accurate. I think, yeah, okay, they pick them up and they eat them, but it's not enough to make a dent in the population. And I do believe that whole initial piece of research got a little bit blown out of proportion. So oftentimes that happens, and we have the same problem with bats and mosquitoes. If you put a whole bunch of mosquitoes in a room and release a bat in there, then the bat's going to eat all the mosquitoes, but that's not really what happens in nature.
Right. And it's kind of an exciting finding and gets a little
Gets put,
traction. Yep.
that's right. Yeah.
And then, so another question is about deer feeding at some bird feeders in a mowed yard. Is that something that will increase the risks of getting ticks in their yard?
Yes. Yes, I do believe so, actually. Some of the work that we have done has shown that the two things in a yard, one is a pile of wood and the other is a bird feeder, do seem to be associated
hmm
with a higher risk. And now that could be that the mice are coming around and eating the seeds too, and the chipmunks, right? I've seen that at my own house, and ticks are dropping off of them. But I feel like the deer also would be contributing to that.
All right. And the last question we have coming in is ticks sensing. Are they primarily sensing just movement, or can they sense CO2 coming out of an animal's breath, or how are they actually detecting their potential carriers?
Yeah, we were just talking about that this week. Like, what are the steps that get them to know? Because they do alert. So I'm sure that they're picking up chemical signals like carbon dioxide. I think they're also getting vibration a little bit too, and so that's helping them to get ready for something because that's their one chance, right? It has to brush by them, and then they're going to grab it. You just got to wonder how many times that happens in nature. Like it feels like since they're just waiting for something to come right
Yeah.
by, that most of the time they're going to miss. So it's the chemical, it's the CO2, and it's for sure a little bit of the vibration as well. That's what gets them alerted and ready to go.
Yeah. Fascinating stuff. Really appreciate all the information. We got a lot of interest from our audience. And again, to everybody on the call, I am going to be sending out all the resources that we got in the chat today. And the recording will be coming to your inbox. So appreciate everybody making time on this beautiful spring night to tune in and learn little bit more about ticks. And really appreciate Susan, all that you shared, and wish everybody a good night.
Okay. Thank you.